Hi, my name is Isaku Tanida of Biosciences. As part of a short series of videos about the value of the AVITI system for single-cell analysis, I'm going to walk you through two important points today. First, the high performance of the AVITI system when combined with 10x Genomics single-cell analysis technologies. And second, a simple cost savings analysis on a per-sample basis using the AVITI 2x75 sequencing kit for single-cell RNA-seq across a typical range of cell numbers and reads per cell. So to begin, let's take a moment to first review how single-cell sequencing has transformed our understanding of biology. The data representations on the slide show the stark contrast when going from bulk sequencing to sequencing individual cells on the right. How the latter uncovers the true heterogeneity of complex tissues such as cancer biopsies or brain. These insights have increased the demand for single-cell applications dramatically and driven the need for solutions to be accessible, scalable, and cost-effective. The next slide will outline some of the important factors that go into designing successful single-cell analysis experiments. When you set up these experiments and the sequencing that's required for them, you need to account for flexibility, to QC your libraries, like sequencing a subset of samples before you sequence your entire project, or to pool multiple libraries together, or to accommodate different single-cell assay types which are growing in variety. And of course, there's throughput. More samples or more cells, or more reads per cell, it adds up quickly as your project evolves. High quality, of course, plays a part. Much like the sample quality itself, the sequencing quality also needs to be high to maximize the biological insights that you can gain from your samples. And of course, cost and accessibility play a large part in the process, where users seek to minimize expense and delay. The AVITI system provides an ideal solution to meet these needs with performance features that make single-cell analysis by NGS more accessible to labs than ever before. Hopefully, you're able to catch the recent announcement of our enhanced sequencing specifications, where the AVITI system now generates 1 billion reads per flow cell. Simply put, 1 billion reads when combined with the $1,080 price of the 2x75 sequencing kit brings the cost per million reads to a level that is unmatched by alternative sequencers. Over the next slides, I'll walk you through a 10x Genomics single-cell sequencing workflow using the AVITI system, and then share an example of cost savings you can achieve across a range of similar single-cell sequencing experiments. In this figure, hopefully you'll recognize this as the 10x single-cell workflow at a very high level. The AVITI system fits in seamlessly, where the completed single-cell library is circularized using a simple process at the bench called ADEPT before loading onto the AVITI sequencer. This circularization process itself only takes 75 minutes and requires no PCR amplification steps. In this experiment, human PBMCs at both 1,000 and 10,000 cells were used separately as starting samples. Libraries were generated with 10x 3' version 3.1 kit and then circularized using the ADEPT library compatibility kit. The libraries were then sequenced on the AVITI system and analyzed and visualized with 10x Cell Ranger and Loop software. You can see in the table the high performance of the AVITI system at both the smaller and larger cell numbers as compared to the requirements for becoming a 10x compatible partner. Through its compatible partner program, 10x Genomics strives to support its customers with validated products from sample collection and sample prep tools to sequencing platforms and data analysis services, all to ensure their customers have confidence in the entire 10x workflow from start to finish. So Element is very proud to be only the second sequencing platform to be listed as a compatible partner as part of their qualification process. So after taking you through an actual single-cell experiment, I want to walk you through the calculations of similar experiments performed on the AVITI system within a range of cell numbers, number of reads, and sample numbers, so you can see what the average sequencing cost per sample would look like as compared to an alternative benchtop solution. You can see in the table that using the 2x75 sequencing kit and the dual flow cell design of the AVITI system, this offers an unmatched combination of cost savings and flexibility versus the alternatives. I hope this review was helpful to understand how easily the AVITI system can plug into your NGS single-cell analysis projects, and the tremendous user advantages and sequencing cost savings the AVITI can mean to you when you're planning your next experiments. A thank you to our partners at 10x Genomics for their collaboration and support. For more details on the performance of the AVITI with the 10x platform, please feel free to download our 10x datasets from the Biosciences website, and look for the upcoming release of our 2x75 sequencing kit, offering the lowest cost per read in the industry. Thank you.